All right, BIM 100 crew, let's talk. The final exam for Professor Pasquinelli's class is coming up fast, and we need a game plan, right? It's time to get that strategy locked in. So let's just call this exam what it is, the final boss. Yeah, it's going to be tough, it's going to be comprehensive, but it is absolutely beautiful. And this little guide right here, think of it as your cheat sheet, your strategy guide to totally crushing it. I'm serious. You've got this. You've been in lecture, you've done the work, all the knowledge is already in your head. The trick now is just applying it the right way. And that's exactly what we're going to do right now. Let's build that framework together. All right, let's jump right in. Level 1, we're calling this the foundational code. You know how it is in video games? Before you can take on the big boss, you have to master the basics. Same thing here. First up, a concept you can pretty much bet is going to show up: melting temperature, or TM. It sounds complicated, but it's not. All it is, it's the exact temperature where half of your double-stranded DNA decides to break up and become single-stranded. That's it. Okay, but why does this matter? Well, it's all about the bonds. Think of GC pairs as being held together with 3 little pieces of superglue, 3 hydrogen bonds. AT pairs, they only get 2. So it's just common sense, right? More GC bonds means the DNA is held together tighter. You're going to need way more energy, more heat to pry it apart. So the big takeaway is this: more GC content equals a higher melting temp. Burn that into your brain. All right, level 2, let's talk regulation and replication. This is where things get really cool. We're going to look at how the cell actually organizes all that DNA. It's like a massive library, and how it makes perfect copies of it. So your DNA isn't just a tangled mess. It's packaged in 2 main ways. First, you've got euchromatin. This is the open for business section of the library. The DNA is loose, accessible, and ready to be read by the cell's machinery. This is where you see acetylation. Then you've got heterochromatin. This is the archives, the do not touch section. The DNA is packed super tight, totally silent, and locked down with methylation. No reading allowed here. Okay, so how do we figure out if a gene is in the open section or the lockdown archives? We use something called a DNase protection assay. It's actually a pretty clever trick. You take this enzyme, DNase, that just loves to chew up DNA, and you unleash it on the cell. If your gene is in that open euchromatin, well, it's a goner. It gets digested. But if it's tucked away safely in heterochromatin, the enzyme can't get to it. So when you run everything on a gel, what do you see? A nice, strong band means your gene survived. It was protected. But if the band is weak or totally gone, that means it was out in the open and got eaten. Next up, replication, making a copy of the entire genome. Think of it like a highly efficient construction crew. First, you've got helicase. This is the demolition expert, the one that unzips the double helix. Then comes primase, the foreman, who lays down a little RNA primer, kind of like marking the spot to start building. And then the main worker, DNA polymerase, comes in and just starts laying down the new bricks, synthesizing the new DNA strand. And this isn't happening at just one spot. It starts at thousands of places called origins of replication. That's the secret to how our cells copy all that DNA so incredibly fast. Okay, let's level up. Level 3, processing and translation. So we've got the DNA blueprint. We've made copies. Now what? Now we actually have to build something with those plans. We have to make the protein. This is basically the cell's factory assembly line. So a new piece of RNA called pre-mRNA, it's not ready to go out into the cell yet. It's got to get dressed up first. It's a 3-step process, and the order is crucial. Step 1, it gets a little protective hat, the 5' cap. Step 2, it gets rid of the junk. All the non-coding parts, the introns, are cut out, and the important parts, the exons, are stitched together. This is splicing. And finally, step 3, it gets a long, stabilizing tail of A's added to the end, the poly-A tail. So remember, cap, splice, tail. It's got to get its hat, its good clothes, and its tail before it can leave the nucleus. All right, so our mature mRNA is now out in the cytoplasm. The ribosome, the protein-making machine, needs to find it and start reading. But where does it start? It's actually super simple. The ribosome just latches on to the 5' end and starts scanning, looking for one specific sequence, AUG. The very first AUG it finds, that's it. That's the start line. It sets the entire reading frame for how the rest of the message gets translated. But here's the thing. Translation isn't just on or off. It's more like a dimmer switch. And it's controlled by these things called RNA binding proteins. Think of them as traffic cops. If a protein binds way up at the beginning in the 5' UTR, it can act like a roadblock. It physically blocks the ribosome from even getting started. Translation shutdown. But if a different protein binds at the end, in the 3' UTR, it can act like a bodyguard. It protects the mRNA from getting broken down, which means it hangs around longer, and you actually end up making more protein from that single message. Okay, this is it. The final level. We've gone over the core concepts. Now it's time for the real test, applying it. This is all about learning to see the patterns in the kinds of questions Professor Pasquinelli likes to ask, so you know exactly what to do when you see them on the exam. All right, let's put this into practice with a classic Pasquinelli-style question. So take a look at this image. What we've got here is a DNA assay, just like we talked about. They're looking at a gene called BCL2 in normal cells and in some tumor cells. You see the lanes marked minus DNAs, those are our controls, and then you see the lanes where they added the enzyme. The big question is, what's the story this gel is telling us? Okay, let's break this down together. Step 1, always look at your control. The minus DNA sleens show you what the band looks like when nothing gets chewed up. That's your 100% signal. Now, let's compare that to the plus DNA sleens. See the normal sample? The band is still pretty strong, right? That tells us the gene is mostly packed up and protected. But then, look at tumor 1. Whoa, the band is almost completely gone. There's only one way that can happen. In that tumor, the BCL2 gene must be in that super open euchromatin state, leaving it totally vulnerable to getting destroyed by the DNAs. Okay, ready for another one? This is another super common question type you'll see. This time it's PCR, and we're looking at a gene you've definitely heard of, RAS, which is often involved in cancer. They're comparing the gene in a patient's healthy tissue versus their tumor tissue. Just focus on patient 1 for a second. What's the difference you see between the healthy lane and the tumor lane? The whole answer is right there in how those bands look. So the logic for PCR gels is really simple. A bigger, brighter band just means there was more DNA to begin with. And if you look at patient 1, that tumor band is way brighter and thicker than the healthy band. That's the textbook definition of gene amplification. It means the tumor cells have made a ton of extra copies of the RAS gene. So when you run PCR, you have way more starting material and you get a much, much stronger product at the end. Simple as that. And that's it. You've made it through the guide. We've covered the basics, the more complex stuff, and even looked at how to tackle the big boss questions. Seriously, you are ready. So let's do a super quick rapid-fire recap of the biggest takeaways. Ready? Melting temp, or TM, it's all about GC content. More GCs, higher TM. DNA assay. If the band disappears, the chromatin was open. PCR gel. A brighter band means more starting DNA, which means amplification. Post-transcriptional mods. You need the cap, the splice, and the tail. And finally, UTR regulation. Binding at the 5' end is a roadblock. Binding at the 3' end is a bodyguard that stabilizes. Got it? Good. Look, you've put in the work all quarter long. You really do know this stuff. You've seen the patterns, you get the logic behind it. The training montage, it's officially over. You've got the knowledge, you've got the strategy. The only thing left to do is walk in there and execute. Now go and absolutely ace this exam.